So the paper is titled Confronting False Discoveries in Single-Cell Differential Expression. First, I wanted to give a little bit of context for those who don't know. Of course, there are many challenges of single-cell differential expression analysis. And I just wanted to talk about baseline, why are they prone to false discoveries? And it's because of this problem that you might have heard about, called the multiple testing problem, and where you're making thousands of pairwise comparisons. And basically, if you know about how p-value works, of course, if you have many of those, then your threshold for false positives will allow for many falsely called genes. And I just add a note here that because we're referring to false discoveries, there's this thing called the false discovery rate, which is basically the number of false positives over the total number of genes called as differentially expressed. Then there are other challenges known like high sparsity and lots of variability. And both of them will be referred to in this paper. I just put the last note, etc., because there are even reviews about the challenges of single cell differential expression analysis that I can point you to at the end of the presentation. Then a bit more of context, because I recognize that it's very important to understand the difference between pseudobulk and single cell methods in order to compare the benefits and shortcomings of each, which is what this article is doing. So pseudobulk more generally refers to groupings of cells. How you do this grouping can vary. And so, for example, if you see in Archer, they refer to pseudobulks as cell groupings of cells, usually derived from individual clusters that correspond to known cell types, whereas the paper is aggregating cells based on biological replicates. But I will distill this difference later on, and the findings of the paper are more generally applied to pseudobulks, no matter how aggregation is made. And so when I was thinking through pseudobulks and trying to understand them, my first question and my first concern was if the whole purpose of doing single cell methods is to preserve this information at the individual cell level, is there some kind of loss of that information when you do pseudobulk? And just to clarify and to what Fiorella helped me understand is that actually pseudobulking preserves that information because the cell groupings are made based on information that was collected at the single cell level. So no need to worry about that. And more generally, this last bullet point here is just saying that the difference between pseudobulk and single cell methods is at the analytical level where statistical inference is performed. So of course, single cell methods perform the statistical inference at the individual cell level, whereas pseudobulk do the statistical inference at the level of groupings of cells. So if we want to get a little bit more granular, this would be, we would have different gene expression matrices where we would perform the statistical inference. Specifically for pseudobulk, as you see at the lower left, we're performing this inference based on aggregates, or where the column data is aggregated across cells of a given type within each biological replicate. So here. Now moving on and starting to talk about the actual article. The first thing they did is that they benchmarked single cell differential expression methods by applying them to data sets where the experimental ground truth is known. Of course, to do this, they first had to establish what ground truth was. And for them, they said that ground truth was going to be matched bulk and single cell RNA-seq performed on the same population of purified cells, exposed to the same perturbations and sequenced in the same laboratories. And they provide this schematic here of what that would look like. They evaluated performance using this measure called area under the concordance curve, which is basically just measuring the agreement between this bulk and single cell RNA-seq. And they use 14 representative statistical methods, which they identified by conducting this literature review of the most widely used methods for analyzing single cell data. So you see here that almost 90% of the studies that they included in this literature review used those representative studies that they will test. And they broadly fall into three main categories. So pseudobulk methods, single cell methods, and mixed model methods, but they will mostly focus on the article on the first two. Then the data sets they used to test them, they found and they selected based on whether or not they had accessibility to these ground truths and whether the scientists had performed this kind of experiment. So what they found when they were doing this, measuring this area under concordance curve for these three kinds of models, was that the top performing methods, what they all had in common is that they all used pseudobulks to call differentially expressed genes. So here in red, these are all the pseudobulk methods, and all the single cell methods fall behind. What this means by some of them having superior performance is that the ones that have inferior performance are calling many more false positives. And in reasoning about this, the authors went back to the challenges of single cell differential expression analysis, and they reasoned that sparsity is one of the biggest drivers of false positives and difficulties of analysis. So they thought that maybe in lowly expressed genes, pseudobulk methods were correcting for this, and that is why they were performing better overall. And to test this, they separated genes based on their level of expression. And they, contrary to their hypothesis, they found that single cell methods, that the biggest difference between pseudobulk and single cell methods was in highly expressed genes. So looking at this row here, that has the biggest difference than the last row here. And these are the pseudobulk methods. And here's the one that I mentioned in the beginning, you could think about as a representative single cell method. So they also found that genes that were more highly expressed were more prone to being identified as differentially expressed by single cell methods, despite having no biological difference, and that this bias of single cell methods would explain their inferior performance overall. The next and obvious following question was, how do pseudobulk methods avoid this bias towards highly expressed genes? And of course, the biggest difference between pseudobulk and single cell methods, like I mentioned in the beginning, is this aggregation procedure. So they wanted to test whether the aggregation procedure was responsible for the superior performance of pseudobulk methods, or if it was something inherent to those methods and separate from the aggregation procedure. So on the left here, you have the test they did to see whether these pseudobulk methods had inherent advantages independent of aggregation. The way they tested for this is by completely ignoring biological replicates, not aggregating the cells at all, and just applying the pseudobulk methods to individual cells. So the column data here would be the cells, not any kind of cell grouping. Then on the left, you'd have the other hypothesis, which is that the aggregation procedure is responsible for the superior performance of pseudobulk methods. And the way they tested this without doing the complete, like just to isolate the aggregation procedure, and is that they shuffled replicates. So each grouping of cells wouldn't be done by biological replicates, but it would have all kinds of replicates within that grouping while still preserving the grouping itself. So like I say here, aggregated random groups of cells to form pseudo replicates. This is another term. So until now we have single cell, pseudo bulk, and pseudo replicates. And sometimes I know it can be mixed up, but in the article, pseudo bulks are different from pseudo replicates, and pseudo replicates actually are more closely thought about to single cell methods than to pseudo bulks. And they actually, the results of both of these tests were that they induced similar decrease in performance, so similar to single cell methods. And this means that they wrongly called a lot of differentially expressed genes, and they saw a reemergence of bias towards highly expressed genes. So on the right, I put the charts or the figures that show this decrease in performance, and in gray you can see the same pseudo bulk methods, but with that modification they did. So either aggregation disabled or the pseudo replicates, which preserved the aggregation, but did not preserve information about biological variants. And on the left is the bias towards the highly expressed genes. So the more highly expressed genes is like the more significance is given when you're comparing, for example, pseudo bulk to aggregation disabled and biological replicates to the pseudo replicates. So you can see how the chart changes towards the end and demonstrating this bias. Okay. And I think the authors could have done a bit more of a better transition towards this, because it was just like a one sentence thing where they were like, oh, we immediately identified that both experiments entailed this loss of information about variance between biological replicates. And this is really the key of the paper. So I personally was more interested in seeing how they got this light bulb, but I wanted to draw this figure because since it's so important to the thesis of the article and their findings, it's important to understand how variance can be represented between biological replicates. So I extended the metaphor used for single cell methods of the fruit salad, which I'm sure all of you are familiar with. And variance between biological replicates in this metaphor would be, and I give credits to Fiorella for sharing this with me, would be, for example, buying an orange from Safeway versus an orange from Whole Foods versus an orange from Trader Joe's. They're all the same oranges, but there can be differences between them. So for example, I put here that a rotten apple, or you might like some oranges from Trader Joe's better than the ones from Safeway. And in the article, they just say that within the same experimental condition, replicates exhibit inherent differences in gene expression. So only focusing on condition A, even though they're biological replicates, there can be differences between them. Okay. And since the authors noticed that both the tests they had done, so the aggregation disabled and the pseudo replicates and single cell methods, something was going on with the variants. They wanted to understand this better, how basically they handled variants in the expression of each gene. And to do this, they compared the variance of the expression of each gene in pseudo bulk versus pseudo replicates. So they didn't do any kind of, they're just doing a pairwise comparison. They forgot about the aggregation disabled and single cell. That's why I said that pseudo replicates in their mind more closely resemble single cell methods. In a data set of bone marrow mononuclear cells stimulated with poly IC12. So what they found was that here on the left is that single cell methods underestimate the variance of gene expression. So circles are the variants identified by pseudobulk methods, and arrowheads are the variants identified by pseudoreplicates. And you can see in each observation, the variance identified by pseudoreplicates is less than the one identified by pseudobulk methods. So they're basically decreasing the variance of gene expression. And this was a supplementary figure, but I also found it very surprising, is that the most highly expressed genes exhibit the largest decreasing variance. So in this area, you can see how this is decreasing. And this would explain the bias towards calling highly expressed genes as differentially expressed by pseudo-replicate methods and by proxy by single cell methods.
One thing that, oh, sorry.
Yeah.
One thing that wasn't super clear to me how to think about is like, by eye, the changes in variance are not like, I mean, they're lower, but they're not like astronomically lower. I never have a good sense of like magnitude of, because usually you express it as like a ratio of the measured quantity, right? So I wasn't sure how to think about like, is this enough? Is this essentially the same? Like,
in the supplementary figures, they have a plot that was like showing the magnitude of the variance. So I think it was comparing delta variance versus delta variance, but I didn't quite understand it. So yeah. And something also that I wanted to put together and really pull apart is that it was clear to the authors that the variance that this loss of information about variance between biological replicates would lead to higher false discoveries, but I wanted to see what were the steps in between. And so this is why I kind of draw out how this unfolds, starting with not taking into account variance between biological replicates. This leads to underestimating the variance of gene expression, which is shown here, which leads to higher sensitivity towards differential expression calling. And that implies that small changes in gene expression will be misattributed to the effect of perturbation, since they don't have that kind of tolerance or to those small changes in variance. And this, I think the authors could have done a better job of just connecting one to the other. So since they were looking at experiments where there were biological perturbations, they wanted to take it even one step further and ask whether these methods would find false discoveries, even in the absence of any biological perturbation. To do this, they stimulated single-cell RNA-seq data, and this is what I mentioned in the beginning, where they varied degrees of heterogeneity between replicates, and then they randomly assigned them to artificial control and treatment conditions. This is, I think, one of the most surprising parts of the paper, because they found that even in the absence of any biological perturbation, single-cell methods actually identified hundreds of differentially expressed genes. So here you can see that even that, for example, edge R, the one with biological replicate, stays the same across between replicate variation. Edge R LRT, but made from pseudo replicates, has many more differentially expressed genes, and the Wilcoxon rank sum test, which is the one that I said was the single cell like representative method, also has many more differentially expressed genes. They also found that the number of differentially expressed genes stays consistent across between replicate variation. So this is why this line stays consistent. Using pseudobulk methods, but not using single cell or pseudo replicate methods, which further supports the inability of these methods to account for biological variation between replicates and the effect this has on the number of genes falsely called as differentially expressed. So this is a central figure to supporting their hypothesis that the pitfall of single cell methods is that they do not account for variation between biological replicates. And so there are many sources of variability in these experiments, as you all know, including genetic background, environment, intensity and timing of the biological perturbation, and sample processing that can lead to false discoveries. But they note that variability between replicates is easier to control in animal studies. So human studies are actually more vulnerable to false discoveries than animal studies. And they wanted to, since the reasoning was that animal studies were more easier to control, they conducted an experiment on mice that had that were I don't know, hurt by spinal cord injury to validate the differential expression calls of each method. And then they use RNA scope probes to corroborate the findings of each method. They found that 83% of genes called by pseudobulk could be corroborated, in contrast to only 23% of genes differentially expressed called by representative single cell methods. So this is a big difference. Before I go to closing thoughts, I also wanted to show that they made this Libra package where it's an R package where you can compare the methods yourself, and you can use it to do calls for differential expression analysis that and compare the validity between them, which I thought was really cool. For some closing thoughts, I think future directions, and since they made a lot of dramatic calls about paradigm shifts and changing already published papers, I think a simpler way of doing this is just to compare existing methods for correcting for false discovery rates and seeing how all of them implement them, if they're implementing the same ones or different, which can't be implemented by single cell methods. That's something that I think maybe they should have mentioned, because I'm not very familiar with those methods, but for the most famous one, like the Benjamini-Hochberg method, they didn't talk about, and I don't know how these different kinds of methods implemented them. And I think the strong point of this paper is that previous papers benchmarked these methods, but they did that using synthetic data, and they didn't really capture the pervasiveness of those false discoveries. Then another important point that this paper brings forth is that it's important to stress like experimental design and other non-computational approaches to control and correct for the variants that I mentioned in the last slide. And some questions I had, for example, when I was looking at other papers that benchmark statistical methods, were that a lot of them use this thing called the ROC curve, and I was curious why this paper hadn't used that tool. Like I mentioned, when they performed the analysis of variance, they only compared pseudoreplicates with pseudobulk. So why just do it on pseudoreplicates and not on the single cell methods? This we already talked about, which is the ground truth. And this is a tool that was shared in the bioinformatics channel, the Gladstone bioinformatics channel, which is used to correct for continuous and categorical covariates, which if you want to take a look at, I put the link here.